Good morning. So I am Michael Young. I'm a scientist at Skapeton's Eye Research Institute, which is part of Harvard Medical School. I've been doing stem cell research for 20 years now. So what I'm going to tell you about today is my work and some of the background in retinal stem cells in the eye that can be used to repair the retina in retinal degenerative diseases like macular degeneration and retinitis pigmentosa. So I'll tell you a story about how we started 20 years ago isolating stem cells from the eyes and brains of mice and rats, and how we've taken that into humans and now into a clinical trial that has been showing some really early progress in restoring vision in patients who are blinded by these diseases. Okay, so a little review of anatomy. What we're talking about today is the retina. So the retina is that very thin sheet of nervous tissue. Remember, the retina is brain tissue that lines the back of the eye, and it captures light through outer segments of photoreceptor cells, rods and cones, and sends that information throughout the retina, eventually touching retinal ganglion cells, which send the information down the optic nerve to the recipient regions in the brain, principally the lateral geniculate and the visual cortex, and that results in the perception of vision. But it all starts in these little structures. Sorry, I'll review the macula first. So, as I mentioned, the back of the eye is lined by nervous tissue, brain tissue that receives light. Almost all of your visual information, reading, watching TV, looking at your phone, takes place in this little tiny spot about the size of an eraser tip called the macula. So the macula is full of one type of retinal neuron. Does anybody know what those cells are? Cones. So it's 100% cones in this region. Now if you look at the rest of the retina, it's usually made up of lots of different cells. So in the back of the retina, you have the black cells, the pigment epithelium. So the pigment epithelium was the first transplant ever performed in humans using embryonic stem cells. Okay? And I'll tell you a little bit more about that work. And then you have these cells, the photoreceptors, rods and cones. Okay? They have a very close relationship with the retinal pigment epithelial cells. And again, in this region here are the nuclei and the cell bodies. And then there is an inner segment here and an outer segment. So the outer segment are stacks of discs, and their job is to capture photons. Okay? So when a photon of light travels through the retina and touches the outer retina, then you have a chemical reaction that takes place and sends these impulses down and again out the optic nerve eventually. Okay? What else here? So this structure here, it's really a potential space called the subretinal space, is between the photoreceptors and the RPE, the retinal pigment epithelial cells. And this is where surgeons go to transplant stem cells in the back of the eye. And it's quite an amazing procedure when you see them enter. I'll show you a video of the first patient who received a transplant in our trial. It's really a remarkable technique. Okay. So why do we want to transplant stem cells into the eye? Mainly because the retina cannot regenerate when damaged for mammals. So fish, frogs, if you damage their retina, to some extent they'll grow a new one. Salamanders certainly grow a new eye when their eye is damaged. If you cut off a finger, they grow a new digit. If they are injured in a fight, they grow back a new tail. They can regenerate greatly. That is lost in higher vertebrates. Okay? So mice and rats, which we study mostly, and humans can't regenerate their retina. And so when you lose your photoreceptors, your rods and cones, these are the diseases you get. These are the diseases that cause loss of photoreceptors. Ganglion cells, which I'm not going to talk about today very much, but these are the cells that are lost in glaucoma, okay, and other optic neuropathies. In diabetes, you lose all your cells. And that's why there have been no transplantation stem cell studies yet for diabetes. Yeah, so just a cartoon highlighting again to really drive the point home. The retina is a complex structure made up of specific layers of cells. But most importantly for today's lecture, it's this interrelationship between the pigment epithelial cells and the photoreceptor cells, okay? Oh, my students like to take lots of space. Okay. So I've mentioned to you retinal degenerative diseases. These are two examples of the diseases we're trying to treat and what it looks like when you have this disease. So these are complex, some are genetic, some are metabolic, some have unknown causes. We know that the biggest risk factor for macular degeneration is smoking. Okay? But these are the leading causes of irreversible blindness. AMD, or age-related macular dystrophy, that should be degeneration, sorry, affects 15 million people in the US, and that's growing as the population ages. So retinitis pigmentosa are almost all genetic diseases that cause loss of rods and cones, principally rods. And although they affect 2 million worldwide, this is known as an orphan disease in the US because it's really only about 50,000 patients every year are diagnosed with retinitis pigmentosa. But the common feature of both of these diseases is you lose your rods and cones, okay? And they don't grow back. So our goal is to try to replace these cells. So just a few features of age-related macular degeneration. If you're over 40, 1.5% of the population has this disease. If you're over 80, this rises into the 20 and 30% range. So most of your grandparents have a good chance of coming down with AMD sometime in their life. If you live long enough, most of the time your eyes degenerate. And there's really interesting data coming out now on why that is. But we're not going to get too much into that now. But again, the cause is not very clear. Something known as the complement pathway, which is an immune inflammation pathway, has been implicated in this disease, and there's some new therapies working on that. But again, trying to make it very clear, the retinal pigment epithelial cone interface is diseased. And what that means is, someday we hope to replace both of these cells with a transplant. But we're not there yet. So different people are transplanting these cells, and we are transplanting cells like this. So one thing that's come along to treat one form of macular degeneration that's been a huge breakthrough are what are known as anti-VEGF treatments. So what that means is blocking blood vessels. So VEGF is vascular endothelial growth factor. Okay, so this is a chemical that's released in your eye when it's diseased. It attracts blood vessels. So if you want light to get to your retina and stimulate the cells, you don't want a lot of blood vessels there. And more to the point, you don't want them leaking. So when you get the wet form of AMD, it's divided into wet and dry. Your blood vessels leak, and you basically get a scab in the back of your eye. And in the old days, they would go in with scissors and tweezers and pull that collection of blood out the back of your eye. You can imagine it's not very good for vision. What's new is this treatment, given by an intraocular injection, has really changed the game, and it's the first real treatment for this form of disease. So it stops those blood vessels from growing. They go away and you see better. So that's the new, what we call standard of care in this disease. But it requires monthly or semi-monthly intraocular injections, and nobody wants a needle in the back of their eye. It's not very pleasant. So we're trying to come up with new forms of drug delivery. So maybe you only have to have an injection twice a year or once a year. So that's some of the new research there. But many people have gotten this. It's known as Avastin. is the drug, and it's very effective. So this is a slightly older slide, but it highlights that there are a tremendous number of clinical trials going on now, trying to treat blindness with stem cells and gene therapy. I won't go too much into this, but I grew up with a lot of these people in the field. Many of us worked together as students many years ago. We've all gone on to do our own thing, but several of us have gotten this into the clinic, and I'll tell you about those trials soon. So the real challenges in terms of treating blindness with stem cell therapy are getting your cells to survive the procedure and the microenvironment in the back of the eye. But once they survive, I'm sure you guys have been taught about stem cells, that they can grow forever. You don't want them to grow forever in the back of your eye. You want them to stop growing. So the big risk factor is tumors. So stem cells can form tumors by nature. So what we're trying to do is pull out the good cells that make retinal tissue and remove the tumor cells. Okay? The amazing thing is we're now 5 years, 6 years into these clinical trials, and there's been almost one example of a small tumor that formed, but hundreds of patients have been treated with these cells of various different kinds, and there's been no tumors formed. Rejection is the other problem. These are foreign tissue grafts. So one can either immunosuppress the patients or purify the cells such that they don't reject. Those are the two options really to try to get these cells to survive. So again, there's lots of different approaches to try to regenerate the retina. Many of you have probably heard about the gene therapy trials that have taken place and have cured blindness in certain diseases. And then there's a lot of cells that can be used. There are electronic chips that can be implanted in the back of your eye that can give you some level of vision. It's not very good, but it's going to get better, as you can imagine, as technology improves. So this is an example from 6-7 years ago of one of the first, actually the first, trial of transplanted stem cells into the back of the eye. And so this is, let me think about this. Oh no, sorry, this is an early animal study that took place before the human trial, but this is a paper published by the UCLA group highlighting the fact that you can transplant human cells into the back of the eye and that they can survive. So, as I mentioned, there are lots of different treatments now available for blindness. What is the appropriate treatment for each patient? That depends upon the stage of degeneration. So these are all the rods and cones in this layer here, okay? So if you have all your rods and cones, or a lot of them, maybe you just want to do a gene therapy to correct the problem, if that's possible. The other option is to preserve them with growth factors, drugs that preserve their life. That's another possibility. As your retina degenerates further and you have fewer and fewer photoreceptors, then we start thinking about stem cell transplantation or chips, electronic eyes. This is a really interesting new field called optogenetics. Has anybody heard about optogenetics? So it's pretty wild. What they've done, so again, your photoreceptors have molecules that when they're stimulated with light, they fire, okay? Bacteria have molecules like that in their systems as well. What scientists have done is taken that bacterial phototransduction molecule and put it into mammalian cells. And when they do that, when those cells when those cells sense light, they fire as well. So when all your rods and cones are gone, maybe you can make your ganglion cells become light sensitive. And that's in early stage clinical trials now as well. The real problem with that approach is there's two problems. You don't have any discs. So the discs, as I mentioned to you earlier, are what capture light and are very good at it. So that's gone. And all those different neurons in the pathway to your ganglion cells are also not part of the system. So you don't have any of that processing taking place. So I think optogenetics and prosthetic chips can give blind patients the ability to sense light, but they're never going to be reading the newspaper. Okay? That's when you need gene therapy or cell replacement. So RP, retinitis pigmentosa, is the other disease we're treating. This is, again, is a rare disease. Lots of different estimates depending upon how you slice them up. But again, this is a young person disease. Kids get it, young adults get it, and you go blind. Typically through rod degeneration. And this is what it looks like. All your peripheral vision is lost, but for a while, central vision is maintained. Okay? And that's because the cones are still there. Okay? Okay, now to the research that we've been doing. So many years ago, almost 30 years ago, when I was an undergraduate student, I worked with a guy who did some work transplanting retina to the brain. So why would you want to do a crazy thing like that? Originally, it was done to study development and plasticity, how connections form, how tissues interact. But something really crazy happened when we did that, and that is the retina made connections to the brain in the retinal regions that should receive retinal input. So how do you study that? Well, we studied it by looking at the pupillary light reflex. So that's when you shine light on your eye, your retina, and your pupil dilates and constricts. So it turned out that these crazy transplants in the back of the brain, when stimulated, hopefully this movie works. Let's see, I might have to come back here. Okay. So, we shine light on the brain and the pupil constricts. So how is that happening? It's happening because the retina in the brain can sense light, transmits that information to the brain, and then the connections to the pupillary muscles are still intact. Okay? So this was really the first example of a brain transplant that could relay meaningful information through synapses. So this was 30 years ago. So what many people said was, let's put these retinal transplants in the right place, put them in the eye, and we'll be able to restore vision. So people tried that, and it didn't work. And it didn't work because this retina, this embryonic retina, could make connections in the brain, but couldn't make connections in the retina. Okay, so it just sat there and was disconnected basically by scar tissue. These were mostly done by putting them into newborn animals, okay, when the brain is much more plastic. If you're going to treat patients with blindness, you need to treat them when they're 70, 80 years old. Plasticity is lost at birth, really, okay? So if you want to achieve reconstruction, something has to change. And what changed was stem cells came along. So when I started my career at Harvard, again 20 years ago, people were just starting to talk about stem cells. Okay? So stem cells really have been around for decades in the field of blood and hematology. So when you hear about a stem cell transplant for cancer, these are blood stem cells. Okay? They can reconstitute the bone marrow through a transplant if you pick out the right cells. But about 20 years ago, people started finding stem cells in other tissues. And then they discovered embryonic stem cells. And soon after that, they discovered induced pluripotent stem cells. And I wish I had some more slides on that, but I'll spend a moment or two telling you the difference between these things. So, have you studied embryonic stem cells a little bit?
They've read, here's kind of a minor read a caption and watched one Khan Academy video about a little bit about stem cell.
Okay.
But a little bit.
So the two principal types of what we call pluripotent stem cells, and that means they can make a whole organism, or at least all the tissues of an organism, I should say, are embryonic stem cells and induced pluripotent stem cells. Okay? So ES, embryonic stem cells, come from very early embryos in the blastocyst stage. You tease these cells out of the embryo, and they grow in culture to form all the tissues of the body. Okay? But you have to destroy an embryo to get these cells. So along comes Dr. Yamanaka from Kyoto in Japan and discovers that if you take 4 genes and stick them into a somatic cell, a skin cell, a blood cell, those cells turn into embryonic stem cells, or something very closely resembling embryonic stem cells. Okay? Any questions about that? Okay. So, these are yet another type of stem cell, brain stem cell. So the brain continues to regenerate throughout life. And I've got to speed up a little bit here. But the question is really, can brain stem cells replace dead cells in the retina? And the answer is sort of. So this is one of the first slides that we obtained by transplanting these green rat stem cells from the hippocampus into the brain, into the retina of rats with a retinal disease. Okay? This slide just shows these green cells in the back of the retina making synaptic connections. This is again an example of a cell transplanted into the back of the retina from brain to eye, looking like a photoreceptor cell, but not really becoming a photoreceptor cell. Lots of pretty pictures, I think they're pretty pictures, but still falling short of cell replacement. So, if you want to make photoreceptors, again, pluripotent cells, embryonic stem cells, and iPS cells, many people are trying this. We're working on it. We don't think that's the best approach. We use fetal retinal progenitor cells from fetal eyes to generate photoreceptor precursors. So this is a complicated slide a little bit, but just showing that there are a lot of issues with cell choice if you want to make a photoreceptor cell for therapy. The key is that you need to make a lot of them to generate enough to treat patients. So we make a lot of them by growing them in special incubators that lower the oxygen tension. Okay, so you have incubators here for growing cells. I'm sure that they have 5% CO2, but normal oxygen. Does anybody know what the normal oxygen tension is? What are we breathing now? 20%. So the atmosphere is about 20%. But it turns out in your body, only certain cells in the lungs ever see 20% oxygen. Everything else is lower. And when you get into nervous tissue, like the eye, the actual oxygen tension is closer to 5%. And so we didn't discover this. A very smart woman from California told us about this. Marie Cheta said, you should try growing your cells at 5% oxygen. That might be a really good way to do it. It gives them the oxygen level that they have in their niche. So what we use is a tri-gas incubator that uses nitrogen gas to flush out the oxygen and lowers it to 5%. And when we do that, the cells used to grow like this. This is basically senescence, we call it. They stop growing. Now they keep growing almost forever. And this really allowed us to start a clinical trial. Without this discovery, we would never have the ability to grow enough cells to treat patients. So how do we know these cells that we're growing, these stem cells, are going to work? Well, you do that by testing them in animal models. So this animal is the RCS dystrophic rat. RCS stands for Royal College of Surgeons. Very proud name these rats have. But they go blind. And so you can inject stem cells, and when asked the question, how well do these rats see, you can actually test vision in a rat. So this is the grafted eye. If you don't do anything or inject buffer, salt solution, they have visual acuity of 0.3. They're blind. If you transplant stem cells into the back of their eye, they get about twice the vision that they had without these transplants. The next thing we did was a pig study, okay? So, most of the time, if you want to show your human cells are going to work in humans, you have to put them into rats and mice and monkeys. Those are called xenografts. And they're not very informative, in my opinion. Foreign tissue like that behaves very differently. So we came up with an idea to do what we call an analogous allogeneic study. Okay? So we grew pig cells just like we grew the human cells and put them in a pig. Okay? That's the best way to see what a human cell will do in a human. When we did that, we made lots of photoreceptors. And we went to the FDA, the Food and Drug Administration, to get approval for these patients' trial. We were able to get the indication of cell replacement based on a pig study, even though we didn't put any human cells in the pig. Okay, so it's a very good approach, and the FDA really likes it nowadays. So this is an example. So we used a green pig. There's a very smart guy out in Missouri who made a green pig along the way to cloning pigs. So you know about green fluorescent protein? Okay, so that's very important in biology. And so this pig has green eyes. The whole body is green if you shine black light on it. But it's so green that even in room light, its snout and its hooves are green. Pretty freaky looking. But the embryos are 100% green. So we take these embryos, take out their eyes, grow stem cells from them, and put them into a normal pig. And then we see these new photoreceptors here. Okay? The cool thing is, these cells make synapses. And we know that because all this green in the synaptic layer of the retina. Okay? So, this is what it looks like when you try to grow enough stem cells to treat all the patients in the U.S. with retinitis pigmentosa. You start with 2 eyes, and you grow them into what we call a seed bank. And you grow them some more into what's known as a working cell bank. But the key result is that we can start with 2 eyes and end up with 900,000 doses. Okay? Pretty remarkable. We don't do that. We're conservative and grow a lot, but we can grow a lot of tissue, a lot of cells from 2 eyes. Okay, so the clinical trial that we're working on now is sponsored by a small company in the UK called Reneuron. It's a great name, I think. We're treating patients with retinitis pigmentosa who have 2200 vision or worse. So of course, with your glasses on, all of you should have 20/20 vision. So these patients have 2200. So they see what you see at 20 feet. Sorry, they see what you see at 200 feet at 20 feet. So imagine you're standing 200 feet from an eye chart trying to read it. That's the kind of vision these patients have. So we're injecting these cells directly into the subretinal space I told you about. We got lots of really good help from the FDA. This is all taking place at Harvard. We've now moved on to a second center in Arizona. But the first patient was treated in 2016. A total of now 19 patients have been treated. But these patients, I think, are the real heroes of this work. They knew that they had no hope, and they volunteered to receive these injections, knowing there were risks, to try to put this work forward. Yeah, this gets a little complicated, but the key I want to show you is this is the example of the surgery on the first patient. anybody's squeamish, you might want to look away. So this is not going to work. The key is when you do these surgeries, they're called three-port pars plana vitrectomy. You have to remember that now. But when you do subretinal surgery in the back of the eye, you make 3 holes in the eye. And here you can, so one of these holes gives you a candelabra, they call it, a light. One is for suction and pressure to keep the eye inflated. And the other one is for all your tools, your scissors, your needles. They take these in and out of the back of the eye. And then they inject the stem cells. And I wish you could see this because it's kind of cool. But you can't. So the key is that they're injecting these cells underneath the back of the retina. And we were all very worried that this was a difficult technique in an eye full of degeneration. No complications in 16 patients, which I think is remarkable, and it tells you that these surgeons are very skilled. So I can tell you the result that was just released a couple of weeks ago, and that is that this actually works. So when we started injecting patients with a little bit of vision, they got back a tremendous amount of vision in weeks. And in a month, they were seeing, this is the eye chart that we did. This should tail down to here. But they were seeing 5 extra lines of vision. So originally, if they could see this line, after the transplant, 1, 2, 3, 4, they could now read all of these lines. So it tells us that these stem cells are not only working, but they're actually making new neurons. I'm confident they're making new neurons. We have no way of knowing this. One problem with stem cell transplantation is once you inject them, they kind of disappear. We have no way of tracing them. So that's a problem. Okay, so I just want to tell you a little bit about where I work. So at the Ocular Regenerative Medicine Institute at Mass Eye and Ear, Harvard Medical School, we started as a retinal transplantation center, and we've grown to have expertise in stem cell biology and regeneration of all ocular tissues. We have 23 labs, if you can believe it, all working in this area, over 100 fellows and students. And so these are the teams of people that worked on, first of all, the pig studies in Denmark that I told you about. And these are my labs over the years. It changes, it gets bigger, it gets smaller, the faces change. But most of this work, importantly, was done by this guy, Peter Baranoff, who's now a scientist at Harvard Medical School at Well. And yeah, so I'll stop now and answer any questions you might have. Questions? Oh, I'll ask it once. Go ahead.
How long did it take for when you started those experiments?
17 years from the first rat study to the first patient. Yeah, it's a long time.
What made you decide to start working on this research?
So I was an undergraduate back in Pittsburgh, trying to find a research project to fill out I knew I wanted to be a scientist, but I didn't know what I wanted to do. And I was looking in this book, and this guy was doing brain transplants. And I went to his office that afternoon and said, "This is the coolest thing I've ever heard of. I want to do this." And that led me into the retina. So I originally started studying the brain. My undergraduate was in behavioral neuroscience, which has kind of become neurobiology, that kind of, neuroscience. But I originally was a brain scientist and then moved into the retina. But of course, is the retina brain? Yes, it is. Right? Okay?
So something they might find interesting that I remember from, because I, over the years, followed the research a bit, the pig study, some of your challenges you had in those phases, remember? And even just getting them on the bed. Remember?
Yeah, it wasn't really until we got to Copenhagen when everything was really organized. So in Denmark, the biggest industry is ham. That's their most important export. So it turns out in each country, wherever you have lots of animals being used, they're used in medical research. So they have expertise in pig studies there. But the problem in some of our early pig studies was, if we wanted to do a long-term study, we had to use a crane. Because you know better than I do here probably, pigs grow very fast. And soon they weigh 1,000 pounds. So we now use mini pigs. There are mini pigs and micro pigs. But we use mini pigs that only grow 40, 60 pounds. We did one study where somebody made a mistake or just didn't tell us. But the mini pigs were bred with normal pigs. And as we're doing this study with these little tiny pigs, we realized you take temperature and activity and body weight measurements periodically. These guys were growing fast. And so if they are mini pigs, even with a normal pig half breeding, they don't get to be 1,000 pounds, but you still need a crane. So now we only use mini pigs. It adds to the cost of the study, and their eyes are a little bit smaller, but not much. The reason we use pigs in our studies is their eyes are very much like human eyes. They don't have a macula that I described to you, but they have an area centralis that's very enriched in cones, but not a macula. Yes?
You mentioned earlier that you think that stem cells are creating neurons.
Say again, a little louder?
Earlier you said that stem cells, when being injected, you think that they're making more neurons.
Yes.
So hypothetically, could that be applied to like treating brain damage?
So I can tell you that the company Reneuron that we're working with right now has a trial for stroke, treating brain injuries with stroke with stem cells. ALS, amyotrophic lateral sclerosis, a horrible disease that kills you rapidly, is being treated with stem cells. Spinal cord injury is being treated with cells that don't make neurons, they make oligodendrocytes, a support cell in the nervous system. So yes, all of these diseases, Parkinson's disease especially, is very amenable to stem cell treatment. Things like Alzheimer's disease, where we really don't know what the neurons are that are targeted, they're harder to use. But I think spinal cord injury, traumatic spinal cord injury, and paralysis, etc., are very amenable to stem cell treatment.
And if you're looking forward to the future, because 17 years back, where do you envision it going, or where do you like it to go?
So yeah, I learned a lot of lessons in that 17 years. And so we started a company to try to commercialize and bring to patients a cone stem cell. It's a little different. It's selected and purified. We hope to do that in about 5 years now, because I've learned a lot about how to do this faster. But it's still, if everything goes fantastically, we're still 5 years away from treating patients with these new cells. And we've only been working on them for a year. So the future really is tissue engineering, where you generate tissue in a dish with things like bioprinting, 3D printers with cells and matrices. That's what's coming in the future. Not just cells injected as a single cell suspension, but tissues used to reconstitute body parts. That's the future, I think. Yes.
You said that they do like electronic chips in the back of the eye.
Yeah.
What is it's not that effective, like it's seeing? What do the chips do that they're not?
So all the chips can do, so the chips, so I can tell you it's amazing. There's like 20 projects in the world trying to stimulate anywhere from remaining photoreceptors all the way back to the visual cortex and every step in between. So the one that's approved is called Argus 2, like the Greek Argus. What it does is stimulates the retina en masse. So it's got electrodes connected to a camera on a pair of glasses. So the pair of glasses relays the information, not via a wire, but infrared, to this electrode array that then stimulates the retina, okay? But it's just a sort of it's not random, but it's not targeted like a photon would be, okay? So that leads to low acuity, okay? So what the company that sells this, and it's an approved therapy now, you get insurance to pay for this, not a clinical trial anymore, it's an approved therapy. They claim that you can recognize shapes
So that's just.
and big letters. So it's very low resolution. But the hope is that those microarrays that have like, I think they have like 250 electrodes on a little patch of retina. The problem, as I see, it's really hard to get an appliance, an electronic device to communicate with the central nervous system. We're getting better at it, but we're not there yet. So hearing, think about a cochlear implant. Somebody who's deaf, they have this device in the side of their head that stimulates their cochlea. Vision is so much more complicated than hearing, okay? Colors, intent, all hearing really has is intensity, okay? That's what's encoded in these devices is loud or soft, okay? Vision is much more complicated. So we're just further behind in terms of, the long answer, but in terms of giving high resolution vision with a chip. It'll happen someday, but we're not there yet. Thank you all very much. Oh, thank you very much. Thank you. Thank you. Thank you very much. Thanks, guys. Thank you.